Okay, it's my great honor to introduce our speaker this morning. Oh, okay, come in, just a second. Yeah, so it's my great honor to introduce our speaker this morning, Dr. Sara Valle, senior scientist and project leader with Decision Applications Division at the Los Alamos National Laboratory. She does amazing work combining internet data with satellite imagery to forecast disease outbreaks. She does a lot of really exciting work in mathematical epidemiology. So she got her PhD from University of Iowa under the supervision of Harvard Health Code and Mark Hyman in 2005, and then went to Los Alamos ever since, and she's won many awards. I'll highlight the one she got just last few days ago, like last week or something, and it's a Global Security Medal, which is one of the highest honors given at the lab, which recognizes the exceptional achievements of active and recently retired employees who have made outstanding contributions to the lab, the global security mission of the lab. So she won the medal just a few days ago. So it's a great honor to have Sara join us remotely and present her lecture. And where's the screen or the title? Okay, yeah. So it's global comparison of COVID-19 variant waves and relationships with clinical and demographic factors. Thank you, Sara.
Thank you so much, Abba, for the introduction. I'm sorry that I'm not there in person, but I've watched some of the videos, and I think a lot of the discussions have been really fruitful and interesting, and so I'm really looking forward to hearing more about what's happening today. So this is a project that we have started, and so I don't know if I'm going to be using a full hour, and so I think there's also a lot of opportunity for feedback. But we've been looking at sequencing data, and I'll go into more detail during my presentation. But basically, the goal is to kind of exploit this data, and hopefully will help us with our forecasting efforts. So I wanted to start with a short introduction about our goal and our vision and why we feel we need sequencing data. As many of you know, epidemic forecasting has kind of started to play a key role in not just during the COVID-19 pandemic response, but for the last 10, 12 years after the H1N1 pandemic, the CDC kind of began to figure out the fact that, they just didn't want models to understand and assess the impact of mitigation strategies, but they also wanted to forecast infectious diseases. And so they've been doing a lot of forecasting initiatives for flu, Zika, chikungunya, dengue, and some other diseases. And so that kind of provided the foundation for COVID. And so when COVID emerged, then there were a lot of groups that were already doing kind of forecasting. And so including our group here at Los Alamos that participated in the flu forecasting and some other forecasting challenges for the CDC. So there's been a lot of forecasting models that can forecast cases, deaths, hospitalizations, but only a few have actually incorporated sequences. And so here, if you look at the figure that I included on my slide, and this is a figure from Nick Reich and others on the predictability of COVID-19, they demonstrated that there were many instances during the COVID-19 pandemic where the models were completely off and the accuracy was very low. And so I've kind of highlighted two major areas, and that's the first one is when the Delta variant emerged. And so most of the models, because there was a significant decrease in number of cases, most of the models were forecasting that the number of cases were going to continue to kind of either stay in a steady state or decline. But we saw a very steep rise in number of cases. And also there's been, other cases where when new variants have emerged, the models haven't really been able to incorporate that change. And so that's one of the major limitations. And so our hypothesis and what we're currently trying to do is see how we can incorporate sequencing information so that we can ultimately improve disease forecasting and increase the accuracy. And right now, I'm only going to be focusing on the, we're kind of just analyzing sequencing data, and we're kind of trying to understand how it will inform forecasting efforts. We still haven't gotten to the forecasting part. And so I'm just going to kind of walk you through some of the analysis that we're currently doing. And so here's a schematic representation or cartoony representation of what we feel we want to do. So we have this sequencing data that is the first kind of box here of that I'm showing here. And obviously that data comes from a country of origin, let's say South Africa or India or some other country. And so that information obviously can inform forecasting efforts for that particular country, for the origin locations, whether South Africa or India. But our goal is to see how we could learn something from this sequencing data in an origin country and then transfer the data to another location. And so basically, if there's something that emerging in South Africa, how can we take that information and forecast cases in other parts of the world, not just in South Africa? And so, so that's basically another part of the motivation, and this is where hopefully we will be going in the future. So for this particular presentation, and some of the things that we've been doing is characterizing past and ongoing variant transitions. We're also trying to explore the associations between variant transition and geographic location characteristics, demographics and mitigation strategies in vaccine and all of that, to see how that is contributing to the spread of the variants. And then finally, we want to explore similarities in various Omicron subvariant transitions across locations to see how we could potentially learn and eventually forecast cases. So here's a summary of some of the data that we are using. And so we basically had to harmonize this data. And so some of the data comes on a daily basis, and some it's like static, such as population or health systems capacity or economics. That's kind of like a static pictured, whether it is provided on every 10 years from the census, or it could be like for economics, sometimes they do quarterly kind of updates. We have obviously clinical surveillance cases and vaccination information, behavioral response, and so this is mandates, and then data from GISAID, and so that's the sequencing data that we're trying to use. And I'll go into a little bit more detail about the spatial resolution, but basically for some, in some cases, we're able to get it at the country level, and in some other cases, we can get it at a more finer resolution, like state level, for example. So here, I just wanted to kind of show you, you've probably seen many of these figures of the different variants that have emerged throughout the pandemic. And so here I'm just showing some of them. And so basically we, for this particular study that I'm going to be showing right now, we analyzed over 11.5 million sequences around the globe. And then obviously each of the sequences, we use the Pango lineages associations that GISAID provides for each of the sequences to kind of to group all the different variants. And so here, I just wanted to kind of illustrate some of the heterogeneity across different locations and why it is important for us to really understand what's going on and how the landscape and the background is playing a role. Because as if you see the variants are across the world, then you're going to see many different different kind of time series. And so here I have 4 different countries. So India, South Africa, Brazil, and the United States. And it probably, I don't know if it's easy or hard to see, but you can see that, there's clearly differences between India and South Africa, for example, in terms of some of the variants that emerged early on. You can see that here in South Africa, you have a significant, I think it's a beta wave, whereas in India, the beta wave was really small. And then here in Brazil, we see a gamma wave too, and we didn't really see that in the United States or in other parts of the world. And then as we move towards the end of the x-axis, you can see also a lot of the Omicron variants that have been spreading, and you can see that in the United States, there's a lot of also variability in terms of the variants compared to the other countries. And so clearly there's differences in timing, magnitude, and speed of transitions for all the variants. And so we are going to try to see if we can understand that aspect. So the way many papers and other people have been modeling variants is basically they just use a multinomial model. And here I'm just kind of summarizing the model, which is here you have the variant proportion. And then we have like multiple categories of generalization of the logistic regression. And then this is a function of time. So Figgins and Bedford have a paper where they use the same approach, and this h of t that we have here in the exponent, they assume that it's linear in t. And if you assume that h of ij is linear in t, then you have very nice interpretations of the growth rates. And so they are able to kind of map the growth rates to an R naught and present a really a very beautiful kind of analysis. But the problem with the linearity is that sometimes the fit of the variance is not very good, and I will show you in just a second how that differs. And so in our analysis, we decided to take a very similar approach, but instead of assuming that h of ij is going to be linear, we decided to make it more flexible. And so we're using a cubic spline model for HIJT. And in this, by changing this assumption, it gives us better fit. But on the downside is that we lose the nice interpretation that Figgins and Bedford actually provided in their paper. And so you gain some things and you lose some others. And so here's why we decided to kind of change the H parameter. And so you can see here on the top where our results using the Hij and assuming that it's linear. And so you can kind of see here that the fits are not very good for that particular variant. And then you can kind of see at the beginning of the pandemic, the fits are not, they don't seem to match very well. But then later on, I think it gets better. But because we wanted to analyze all the variants that have emerged, then we decided to just use the more flexible Hij, and so you can kind of see that the fits are better. Okay, so now, now given that we have a model, and so we decided to kind of summarize the transitions of the variants in using 3 different metrics. So one is the speed, or is the maximum slope of each variant. The second summary metric is timing, and so timing of maximum slope relative to a global minimum. And then finally, the magnitude, or this is kind of like maximum height or prevalence of the variant. And we only model and summarize locations with sufficient variant data. And this is, and I'll show in just a second why, but this is because not all the countries are sequencing at the same rate. And so then if you don't have enough data, then it's going to be hard for us to fit the model and to have a more robust kind of solution. And so here I'm kind of showing you all the locations with sufficient data that we use. So basically we have 215 locations out of 590 locations. And when I say, for example, Australia 6, we are doing Australia in 6 regions within Australia that are providing information. Same for like the United States. We have 51 states also, in addition to the country level information. And so now I'm just going to kind of jump into each of these different metrics: the speed, the timing, and the magnitude or prevalence, and then show you the differences that we have been able to capture through this analysis. So here is the speed or the slope of the transition. And in this figure, you can see that in the y-axis, I have all the different variants. So starting from alpha, you go all the way down to Omicron. And then on the x-axis, I have the maximum slope. And basically, if farther from the center, it's faster transition, and then you have the slower transition on the left side. And so you can see all these 215 regions that we have are kind of spread out across the each of the different variants. And I should also say that there may be some variants where maybe we don't have all their regions because they didn't have enough data, or because that particular variant probably didn't spread very much in certain regions. But here, if you look at the graph, you can see clear differences across the variants, and the speed basically is very different across the world. And there's a lot of variation, and that we obviously hypothesize that is because there's variant competition and immunity that is contributing to the slope. And so here I have also we have labeled some of the countries that were the fastest in terms of transition for that particular variant. And so that can be kind of interesting when we are trying to transfer this information. And so now this is the same information that I was just showing in the previous slide, but now I have it in maps. And so we have here alpha, gamma, delta, Omicron BA1, Omicron BA11, and Omicron BA2. And the darker the color, the faster the slope and or the speed of transition, and then the lighter is the slower the transition. And so you can clearly kind of see here the heterogeneity across the world for each of the different variants that we're analyzing.
Question, Sarah, here. It seems like there is this BA1 that has been wreaking havoc in Greenland. Is that true?
In Greenland. Oh, in here, you mean right here?
That's right.
Well, that's a really good question, Greg. Because I don't think I saw Greenland in the list of countries that we had included. So I need to go back and look at why Greenland is. It's
maybe all people who live in Greenland got infected at the same time, and that was it.
Yeah, but I think they wouldn't. I mean, if it's only like a handful, because obviously there's not a lot. The population is. I don't think it would have gone through our exclusion and inclusion criteria, because it was just. We are trying to get only locations that have enough data so that our analyses are more robust. So I have to look into this, but thanks, Greg, for pointing that out. Okay, so the next metric that we're trying to capture is timing of transition. And so here we, it's a very similar graph as the one that I was showing earlier, but here, we have on the y-axis the different variants, and on the x-axis we have timing of maximum slope relative to the global minimum in days. And so here you can see that this is earlier transition, basically in the countries that are labeled, which is where they were able to, where the different variants were able to kind of take over. And so you can see shorter times for Omicron variants. And so they're right here, and short times for also some of the mu, epsilon, iota due to limited spread. And so now, so here's the same, well, not the same, but as a map of the timing of transition. And I see green line again, Greg, and so I will look into that. And so here you can also see the relative timing of the maximum slope. In here, the darker means that it was the variants were able to kind of take over faster, whereas with the lighter one, it took longer for the variants to kind of take over those regions. Let's see. And so then the third metric that we've been interested in kind of characterizing is maximum prevalence. And here you can, it's a similar graph. You have the beta, the different variants, and then we have on the x-axis the maximum variant prevalence. And so there's also a lot of heterogeneity, but there's also some kind of trends that you can see. And so you can see that beta, gamma, mu, epsilon, and iota were generally associated with like lower variant prevalence, and you can kind of see them clustered around here, kind of closer to the left. And the maximum prevalence had little heterogeneity across locations for Delta. So here's the Delta variant. It was, quite separated from the other variants. And then we have like the Omicrons, which have been kind of all over the place. And yeah, and so now I'm just going to show you the map of the maximum prevalence. And so you can also see that we have all these different variants, and the prevalence obviously for each of them was very different across the different parts of the world. So so other things that we've been kind of trying to, we've been doing different types of analysis to just see how we are going to be able to incorporate this sequencing data into epidemic models and into forecasting models. And so here, one of the hypotheses that we had also was, what is the impact of co-circulating variants and how they may be influencing the slope or the magnitude or just the timing of the variants to transition or to take over. And so you can, here I have on the x-axis, I have the number of the variants. Basically, you can see alpha, beta, and also the number of co-circulating variants. You have for, we kind of cluster them by 2, 3, and 4. And so those are the different colors that I'm showing here. And you can see that a higher number of co-circulating variants was usually related to lower transition slopes. So usually the darker ones, you can kind of see that in general, the more variants you have in your region or your country, it's going to be kind of harder for the new variant to kind of take over. And so that's why it's going to be a lower transition, just because they're kind of competing for susceptible people. And so in that, there also, what was kind of interesting is, you kind of have all these original variants kind of cluster around here, and then Delta came, and you see a very significant kind of shift in the maximum slope and just in how they have been kind of behaving. And so this is some of the impact of co-circulating variants. So I think if we one of the lessons learned from this particular analysis is that if there's too many co-circulating variants in a region, then that could tell us something about the transition, and that will help our forecasting efforts. Another thing that we wanted to analyze is what is the impact of vaccination, because we know that vaccination is going to play a role, and if you have regions that are highly vaccinated, maybe there's going to be some kind of differences in how the different variants are actually spreading. And so so here I have the transition timing, and then here I have the transition speed. And so you can see that on the x-axis, I have the people fully vaccinated per 100,000, per 100. And then on the y-axis, on the first one is the transition timing, and then on the figure on the right is the transition speed. And so you can see that kind of higher vaccination rates are associated with lowered in later variant waves. But obviously that changed once Omicron emerged, because the vaccine was only targeted for Delta and previous variants. Then we saw a reduction in the impact of vaccines in slowing the rates, the speed of the transitions. And so Delta also was the inflection point, and you were able to see that in the previous figure where most of the variants kind of behave similar, and then when Delta emerged, then we saw very different dynamics. And here in this analysis, we're also seeing that. And the way we perform these are just pairwise Spearman correlation associations that we did for each of the variants, and then vaccination rates. And so we also decided to do to use a random forest to see the associations and the adjusted associations for variants and all the different data that we had collected, and how everything is kind of working together in terms of the landscape. And interestingly, we found the vaccination association persisting for all, for the transitions, for the maximum slope, the timing, and also the prevalence. And you can kind of see there the driver that had the most impact was vaccination, and that's although in the bottom. And then we have the second most impactful is the number of co-circulating variants. And then the next one is the cumulative cases per million. And we know that obviously the number of cumulative cases per million has something to do with natural immunity. And so this is important, so that we know that if we take those three ingredients into consideration, they will be able to kind of help us assess how a new variant will potentially spread in a new location. And then another, these are just some coefficient estimates for the regression modeling that we did. And so here you can also see that the impact of, here's pre-Delta vaccinated, Delta vaccinated, and Omicron vaccinated. And obviously, because this is before the new Omicron vaccine was released, this analysis, you can see that the vaccine has no association in impacting Omicron, but it does have an impact in terms of reducing, having a positive association with Delta, and also the pre-Delta vaccinated. And so, okay, so here's some other kind of analysis that we've been exploring, and these are just correlations across waves. And here I have Texas and New York. And so lots of competition. We wanted to kind of look at Omicron waves just because that's the most current variant that is circulating. And so clearly there are clear differences in these two states, even though they're both in the United States. But we know that it has to do with the landscape and the number of people vaccinated and the co-circulated variants and everything. And so what we did notice was that older transitions were less relevant. And so now that if we wanted to understand just Omicron, that we needed to just kind of look at the most recent kind of past, because that was going to be more important for the immunity than previous variants. And so the last kind of analysis that we did is clustering. And so we cluster locations in terms of Omicron variants in transition similarities, because we want to start learning what are, if different countries have similar landscapes, if they are having a similar outcome, basically. So here, and on the map here, you can see kind of like there's a cluster in kind of green, orange, purple, pink, and green. And the clusters 2 and 3 are more similar, and the clusters 4 and 5 are very different. And you have South Africa and India, and those two countries are the ones where new variants have emerged, and they seem to have very different kind of dynamics. But then once the variants start spreading, there are some regions that actually seem to be behaving similarly based on their landscape. And so here you can kind of see the United States and Canada, and there's some also similarities with Europe. And then there's this orange kind of clusters that we've observed. This is another thing, which the pre-Omicron clusters were very different. And so these similarities will be useful for cross-location forecasting, especially if a new emerging variant is kind of affecting one country, we can kind of use that information to inform another country. And so I guess some kind of key takeaways from the analysis that we've been doing, and like as I mentioned, we're still kind of in the middle of this analysis, and we haven't really been able to transfer it to forecasting, but that's where we're going. And so we compared different variant transitions across the globe, and we tested a very simple, flexible strategy for modeling many co-circulating variants. And we identified some associations between location characteristics and variant transitions, in particular, vaccine, co-circulating variants, and natural immunity seem to play a role. And although intuitively we knew that, I think quantifying it's more powerful. And so we also did some clustering analysis to see how we can use some of this cross-location knowledge transfer, and because our ultimate goal obviously is to forecast cases, but we don't want to wait until the variant is already emerging in the new location. We want to do it earlier on, and so that's why I think learning from other countries would be really powerful. And so the next steps obviously are to kind of take these relationships that we've been kind of exploring and assessing between sequencing and cases, and see how we can ultimately forecast cases. And so this one is kind of just showing again the vision that we have in our team, which is to take variant information and hopefully that could inform forecasting. And I think that's all I have. So if you have any questions, I'm happy to answer questions.
Excellent. That's a fantastic presentation. Any questions here? Oh, Greg, of course.
So, Sarah, do you have some thoughts about strategies that
might be useful in identifying variants beyond the type of, I think, reasonable and straightforward approaches that you guys have already tried? Is there a way to monitor maybe different parts of the world more specifically? Are there any patterns that you can sort of think of based on, so for instance, we've been talking about it a little bit yesterday. If we had ability to get perfect data, what would be the type of data that one would need in order to be able to sort of look at the kinds of questions that your team was looking at?
Mm-hmm. So that's a good question. And so here is the, there. So here's the map that I feel like would help me answer your question, Greg. And so there, as you know, there are so many parts around the globe that are not really doing good at sequencing, and the data is very poor, sparse, or sometimes it just, I think there a lot of the sequences that are also submitted have errors, and so in the way they're being processed, or I don't really know exactly why there are many errors, and then eventually you can't really use them because they don't really have all the information. And so I think if we want to really, we know that variants can emerge everywhere, especially in areas that are not vaccinated, because those areas, they're more prime for the virus to mutate and develop and new variants to emerge. And so I think if we want to better model and forecast cases, or then we need to start collecting information in all these red areas, because we are only looking at like a very small part, I guess, of what's happening around the globe. And I think that there's more opportunity for increasing surveillance systems. Maybe, I don't know if Bill Gates or Elon Musk can pour some money into increasing surveillance system. But I mean, I think if you look at this map, I think it looks pretty sad.
I agree with that, and I'm really glad you made this point, Sarah. This is excellent. The need for large-scale genomic surveillance. The reason we knew about Omicron was because of the world-class surveillance, genomic surveillance done in South Africa. We need a lot more countries to be doing that. I think countries like Germany, the United Kingdom, Israel have been doing a lot of that in South Africa. But we need to do it across the board. Otherwise, we'll not be able to detect new variants as they are emerging. And that's a big problem. But I have a question for you. So if a new variant emerges into a location with already multiple variants, like when Omicron came, Delta was already in town, and maybe a few others that were somewhat suppressed by Delta, but you say the likelihood of a new variant to somehow compete and survive is dependent on how many other co-circulating variants are. But is it also somewhat dependent on the new variants' transmissibility? It's R0, for example, because essentially what's happening is that they figure out a way to coexist, or there's competitive exclusion. So is it related somewhat, if you calculate the R0 for each of the variants, you compare them, is it likely that the one that has higher R0 will definitely display the others, or so the data shows that maybe is the data showing that some kind of hierarchical likelihood of new variant displacing others based on its R0 or based on its transmission capacity?
So I do think that there, let me just see if I can, I think I've had maybe here that you do see, I think that the variants obviously that have more, that are better able to spread, obviously I think will be the one to win. But you still see, so for example, if I take here March 22nd, you see these two kind of variants kind of crossing. And so there's significant like spread for both of those variants at the same time. And obviously this variant is kind of coming down and the other one is taking off. But I don't know, I feel like that the previous belief that we had in the modeling community that, you know, always one will emerge the victorious, or the one is the one to spread more. I think that it does work in some cases, but I feel like COVID-19 has shown us that things can change very differently. And I mean, I think there's so many things that we learned from COVID-19. And one of them is I feel like you can have like here two kind of variants taking over, like this kind of dark blue and another even darker blue. They're both like spreading almost, you know, simultaneously, and you don't really have one with, I mean, even I don't know what they are, not what's for these two, but that they both somehow can coexist and survive and spread. And so so I'm not sure that I'm answering your question, Abba, but I feel like that there's a lot more that we need to dig in to understand.
You answered my question. Actually, I was motivated by a paper you wrote a long time ago on multiple strains with FEG. You may remember a science paper where you had the coexistence and competitive exclusions. And that's what kept in mind when I was seeing this. Anyway, any more questions? Yes.
Thank you very much for your excellent talk. So in fact, I have two questions. My first question is, you talk about sequencing at the beginning of your talk. And finally, I have the impression that sequencing only serves to identify variants, but do you have other information of sequencing? For instance, sometimes phylogenetic trees, if they are available, they can give also information on transmission rates and speed of transition. And my second question is, everything seems to be model-free because also you did clustering, etc. But to fulfill the program that you have given, are you going to somehow use some model at some point, or the purpose was really to remain model-free and just use data methods from data sciences? Thank you.
Yeah, so, okay, let me answer your first question about the sequences. And, you know, there's all these like phylogenetic tree analysis that the immunologists are working on. And so some of the people in my team are people that work with sequences and are immunologists. And so they understand that. And so we are looking at some of that information, but for this particular analysis, we didn't really look at the at any of the information that came from the trees. We just kind of decided to explore it from a more, you know, using like the multinomial model approach. I think eventually, so our goal is to eventually have a model. And so when we start modeling the spread, when we're transition to cases, we will have, you know, the traditional models that are SIER type approaches. Some of the models that we developed in the past are inspired by the SIR type models, but we typically use like Bayesian models, just Bayesian hierarchical models, just because we are forecasting and not just necessarily doing scenario projection. In those models, you can incorporate, you know, the beta and behavior and vaccine, and so they do kind of are more mechanistic in nature. And so our goal is to move to the model and mechanistic type approaches, and I just stay within this multinomial, which is a very simplistic way to assess the sequences. And I think once we start going into the modeling, I think we will be kind of maybe teasing out a little bit more information from the immunologists about identifiability, because, you know, if you have, for example, is a variant more transmissible just because it's you know, it's more transmissible, or is it, or because the vaccine maybe is not working against it, or and vice versa. And so we will probably have to get more information from the people that are doing more of the analyzing the sequences and figuring out how the mutations contribute to the spread or contribute to vaccine avoidance and that kind of stuff. And so I hope I answer your question.
Yes, thank you very much.
Sara, I'm Jorge Velasco. Thank you for your talk.
Oh, hi, Jorge.
Hi. I have a question on your first map. You have 215. Right. That's a very, very, very interesting map. One of the things that I think are very interesting these days is that these boundaries between this green and the red countries, because that identifies in this map lack of data, but probably this lack of data is also indicative of different ways of dealing with the pandemic, right? In terms of density of control, sampling, all the things, vaccination and stuff. And so I wonder, for example, the border between Mexico and the United States, that is a very interesting interface. It's a territory in which you have two different impacts of measurements, intensity of measurements, and probably this is playing a role in the variants, the changes in the variants that we have, the speed at which they grow, when they grow, and the impact that vaccination has on these particular interfaces, because as has been mentioned, variants may arise in this interface area. So I wonder if you have more specific data or have some emphasis specifically on the border regions in the world. The Mexico-United States, a clear one, very close to us, but in this map, you can see several of them that are very important because of precisely of this inequality in the way the disease is being treated. So I wonder if you have some information or ideas on what is happening there. Thank you.
Hi, Jorge. That's a really good question. And I actually don't have data, but I feel like that's something that I would want to pursue because it's such a, I feel like that's a low-hanging fruit that we could utilize to hopefully persuade people to collect more data. But I think that those border regions and, and as you mentioned it, like you can see them all over the world, would be very important. And perhaps you may be able to capture some information, even if you don't have, so I'm thinking like at the border of Mexico that we may be able to capture some of that because of the connectivity with the southern part of the United States. And maybe we can inform what's happening on the other side of the border by surveilling, like in the south. So I think that that's an interesting idea that we can kind of take a look at and see if we can do something about that.
That's very important. And similar also to look at Southern Africa, they are the next door to Malawi, Botswana, and so on. Red, and so yeah, I think that's very good. But I have okay. Christian has a question.
Okay. Hi, Sara. This is Christian Borgs. I have sort of a more global question. So how far are we from a dream that, so we saw, let's say, Omicron appearing in South Africa, and then people said, okay, it will appear in Europe soon and maybe in the US, but we were not really good in predicting it. Or let's say we saw that BA4 and 5 are really taking off in Italy in the summer. And how far are we from saying, well, given the data we have and the data about both the biological data, your sequencing data, and maybe some surveillance data we have in New York and California, how far are we from predicting, oh, it probably will in New York sort of peak at a certain number a month later, and in California, it won't peak that much. So do we have any hope that at some point we are good in this kind of forecasting or not?
Well, that's what we're trying. So I hope that we will get there. And so our hypothesis and that we hope to prove is that by understanding the landscape, we will be able to say, this is the level of, this is the prevalence that you're going to see in New York. This is how fast it may take over from the current variant. And these are, cases and everything. And so we are hopeful. The one thing that worries me is that surveillance systems are kind of slowly dying now because people are just too tired of COVID-19. And so if we don't continue to sequence and to kind of track cases, we're not really going to be able to validate our models and see if they're accurate. And so if we can't validate the models, then it's going to be hard for us to believe in them and have robust solutions. And so if we continue to collect data and good data, I think that we could eventually get there, but I don't know exactly when. I hope soon, but I don't know.
So you would think if sort of this dies down at some point in 5 years, we get a worse new epidemic, we might actually be there that we can predict?
I think at least for COVID-19, I feel like we have a path forward. I don't know if for other diseases we will have a path forward. The other also ingredient, which is very important in that, is behavior, human behavior. And right now we're not really collecting a lot of human behavior. There's no like centralized databases collecting human behavior. And so unlike, we've been always comparing disease forecasting with weather forecasting, and the benefit of weather forecasting is that the weather doesn't care if you bring an umbrella or not. But for infectious diseases, it does. Our behavior plays a big role, and so not just understanding the landscape in terms of the immunity and co-circulating variants, but also if we don't have a good assessment of behavior, then that's also going to limit our ability to forecast accurately. I think we can forecast something, but I don't know how accurate those forecasts might be. And so I think I think we may be able to kind of move the field forward. I don't know that it's going to be 100%.
Let me ask this question differently. A few of us have been sitting together and asking, can we sort of look back and see, well, what would we need in tools for the next epidemic, right? So why didn't we do that well for COVID in the beginning? And what would we want the scientific community to develop in the next few years so that we better equipped? And one of the things you're mentioning is probably better implementing human behavior in some way into our models. I mean, your SIER model, you want to fit this to, you didn't talk about an SEIR plus behavior model, which we probably need, right?
Oh, yeah. I 100%. And so I think we need more models that are able to capture the complexity of the real world, and one of it is behavior. Most of our models didn't incorporate sequencing or these different variants. There's been some models with like multiple strains. It's usually like 2, and now we're in this landscape where we have like 20 things spreading at the same time. And so yeah, we definitely need models that are able to capture the complex dynamics that we're living in. And also, obviously, there's so many other things about communication and maybe like consistency of mitigation strategies. As in the US, like Texas was doing one thing, New Mexico was doing another thing, and we're like right next to each other. And I think when you have policies that are very different, and that you have these borders and people are moving back and forth, I think that I think it was a recipe for a disaster, I guess. And so that's why we struggle a lot with not having kind of consistent policies that would benefit the public health.
I agree with that, Sarah. Yeah, I agree completely with that. So yesterday, I think that one of the themes of yesterday's sessions here at this workshop was behavior, information diffusion, and all that. So we had a really good discussion on all of this, how to incorporate human behavior, and information diffusion, what kind of algorithms and models are being used, and also the policy issue you mentioned. That's great. We talked about meta population models, for example, and different states, different jurisdictions have different policies. Sometimes that could be very detrimental in controlling this. But anyway, thank you so very much. Any more questions before we thank Sarah one more time for a fantastic talk? Very nice. Thank you so much, Sarah, for really excellent work.
Thank you, Ava. Thank you, everyone.
Okay, bye-bye. All right. We'll take a half an hour break and we'll come back to Alan Lloyd, sitting at the back very quietly.
Excellent.